Hello, everyone, and thank you so much for joining our webinar today. We're excited to present to you our speaker today, Dr. Charlene Velichko, who is the Director of Research Biology for the BioMAP Phenotypic Profiling Arm of DiscoverX. Today, Dr. Velichko will be speaking to you about how to use a combination of target-based and phenotypic-based profiling methods to find new indications for current drugs. Today's webinar is being recorded, and your phone line will be muted during the presentation. If you have any questions at all, please enter them into the QA box or chat box during the webinar, and Dr. Velichko will answer your questions following the presentation. Also, just as a reminder, the slides and recording of this webinar will be emailed to you tomorrow to all of the registrants of this webinar. So now, without any further ado, I'd like to hand things over to our speaker today, Dr. Charlene Velichko.
Thank you for the introduction. So there's been much discussion around target-based versus phenotypic approaches to drug discovery, and my message today is that it doesn't have to be one or the other, but in fact the two methods are quite complementary. So today I'll be showing you an example of how information taken from both approaches can be used to prioritize known drugs for repurposing for a specific indication. So the cost of bringing a new drug to market today is astronomical, estimated to be on average well north of $2 billion. Additionally, the failure rate is quite high. Only 15% of compounds that enter phase 2 clinical trials are ultimately approved. So with that in mind, drug repurposing programs are an attractive way to either expand the indication space for an existing approved drug, this is also known as repositioning, or salvage compounds that have failed clinical trials for the primary indication. In addition to expanding the number of patients that could potentially benefit from these drugs, the time to approval is generally much shorter, a 3 to 5 year range compared to a 10 to 20 year range for new chemical entities, partially due to an already established safety profile. So launching a new drug repurposing or repositioning program starts with indication selection. Although several successful drug repurposing programs have been based on serendipitous discovery or by pursuing disease indications with highly similar pathways to the approved indication, for example, psoriatic arthritis and rheumatoid arthritis, evidence-based guidance on indication selection can help streamline this decision-making process and expand the pool of potential therapeutic areas, thus increasing the chances of clinical success. So many diseases have a complex pathophysiology. An example of this is idiopathic pulmonary fibrosis, otherwise known as IPF. IPF is characterized by a progressive scarring of lung tissue, which eventually leads to the inability to exchange oxygen. And years of academic research have identified many of the molecular targets or pathways known to play a role in IPF. So this includes TGF-beta signaling, ER stress, and macrophage biology, and these have all been well documented as potential disease drivers, but it's unlikely that we have all the pieces to the puzzle. And like a three-dimensional puzzle, each piece influences how the other pieces will fit in and determines the overall final shape of the puzzle. Target-based approaches focus on these known pieces without a full understanding of a drug's impact on the larger puzzle. In addition, single target-based approaches often do not take into account the potential benefit of polypharmacology, which may make significant impacts on a multidimensional disease such as IPF. So in fact, the two approved treatments for IPF, which are pirfenidone and nintedanib, are thought to be polypharmacologic in nature. The phenotypic approaches allow what is known about the disease to define the three-dimensional shape of the puzzle. By combining phenotypic and target-based approaches, one can help refine and characterize the pieces of the puzzle. So the BioMAP platform consists of human primary cells that are co-cultured in vitro and stimulated to model complex disease biology. These cultures, which we call BioMAP systems, are initially validated using multiple drugs or tool compounds, and then the culture conditions and the readouts are locked down and tested repeatedly to show robustness and reproducibility. The systems are grouped together in panels. The effects of compounds different clinically and biologically relevant protein-based readouts are measured across multiple concentrations and displayed as a profile. And this profile essentially serves as a phenotypic fingerprint for the compound. So the BioMAP platform incorporates a suite of tools that can be used to address the needs of drug repurposing programs. The main tools are our panels, which cover broad biology, allowing for the simultaneous evaluation of compound effects on diverse tissue and disease biology, as well as disease-focused panels for fibrosis, oncology, and autoimmune disease. Because we run these systems in a robust and reproducible manner, it's allowed us to build a large reference database of BioMAP profiles. We've been able to develop a rich knowledge base around this database and a suite of informatics tools to interrogate the data. So, an example of this are the signatures of safety and toxicity that we've been able to develop based on BioMAP profiles of compounds that have failed clinical development due to adverse effects. And I won't be able to touch on this today, but this platform is also useful for the systematic evaluation of drug combinations, and a combinatorial approach is being heavily investigated for drug repurposing programs. As I mentioned, we've built a database of profiles from about 4,500 reference compounds. This database contains approved and failed drugs, as well as tool compounds that are both small molecules as well as biologics, representing many different target mechanisms. This information is useful in identifying potential mechanism of action for an unknown compound, comparing activities to gold standard drugs for a particular indication, and for establishing biomarker profiles associated with efficacy and safety. So our goal was to use a fairly straightforward approach to identify known drugs that may have a potential as an IPF treatment. The first step was to determine the phenotypic signature of the two existing approved IPF therapeutics, pirfenidone and nintedanib, and to define their shared effects, if any, as a signature of clinically successful IPF therapeutics. So to go back to our original analogy of the puzzle, we're using these two drugs to outline the shape of the puzzle. As I mentioned, these two drugs are thought to be polypharmacologic in nature, and the target or targets of pirfenidone are not completely known. Interestingly, this molecule was originally identified as a potential treatment for sepsis. Nintedanib is a multi-tyrosine kinase inhibitor, best described as inhibiting the signaling of angiokinases such as VEGF, PDGF. Outside of the U.S., nintedanib has also been approved for the treatment of non-small cell lung cancer, and pirfenidone and nintedanib are not thought to share any targets. But assuming that they do share some phenotypic activity, we then would use a shared signature as a scorecard to systematically score the compound profiles of known drugs in our reference database in order to potentially identify novel drugs or target classes that share the same signature. We would then evaluate the activity of these compounds in a separate BioMAP panel of systems that specifically interrogates fibrosis-related disease biology, and then rank the drugs based on their overall potential. So this is the profile of pirfenidone in our BioMAP Diversity Plus panel, which covers a broad range of primary cell biology. So let me just take a moment to walk you through the profiles. There's quite a lot of data shown here. So pirfenidone was profiled at 4 different concentrations, denoted by the 4 different colored lines, and a total of 12 BioMAP systems, which are denoted by the circular icons along the top and separated by vertical lines. These systems cover broad disease biology, including vascular, epithelial, and smooth muscle inflammation, immune cell activation, and tissue remodeling biology. And the drug effects on 148 readouts are measured across the 12 systems and displayed as a ratioed value to a relevant vehicle control that's run in the same assay. We annotate activities on the plot if they meet a defined set of criteria, and the gray shaded area in the middle is a significance envelope generated from historical vehicle control data and is one of the components used to determine active readouts. So, as I mentioned previously, pirfenidone's exact target or targets are unknown, but what this profile tells us is that pirfenidone has anti-inflammatory effects, particularly on immune cells. Pirfenidone also suppresses the proliferation of both B and T cells. We also see inhibitory effects on matrix remodeling biology, including decreased production of extracellular matrix components such as fibrillar collagens, matrix metalloproteases, and PI1. And many of these activities are consistent with what's been previously published for pirfenidone. So this is a profile of nintedanib run in the same panel. Nintedanib is known to target multiple tyrosine kinases, and its pleiotropic effects are reflected in its profile. So similar to pirfenidone, nintedanib shares anti-inflammatory and immune suppressive effects in systems containing immune cells, as well as decreased expression of matrix components. However, in addition to these activities, nintedanib inhibits the proliferation of a broader range of non-immune cells. So, since nintedanib has also been approved for the treatment of non-small cell lung cancer, these broad anti-proliferative effects are consistent with that oncology indication. Nintedanib also modulates tissue factor and thrombomodulin, factors that are involved in the coagulation cascade, which play a role in angiogenesis, also consistent with oncology indications. So when we overlaid the profiles of the two molecules together with nintedanib in pink and pirfenidone in black, we identified a number of shared activities which are annotated on the plot. So many of these shared activities have previously been associated with clinical outcome in IPF in published studies, while others have not. So we then took those shared activities and turned them into a disease signature scorecard, which we used to score the profiles in our database in order to identify compounds with similar effects. So the scoring algorithm that we used was very simplistic. Most of the readouts were assigned a score of 1, with certain readouts that are well known to be associated with IPF clinical activity being more heavily weighted with a score of 2. So you can imagine that we could have expanded our initial scoring algorithm to penalize profiles that have phenotypic signatures associated with clinical adverse effects, but for this first pass, we wanted to keep everything very straightforward. We then scored all of the profiles in our database and ranked the corresponding compounds by score. So this table lists some of the top scores from the database. So the first thing that I would like to point out is that this list includes inhibitors and agonists covering multiple target classes, indicating that there are many potential routes to clinical efficacy. Interestingly, gilgenomycin and radicocol, both HSP90 inhibitors, were the top scoring compounds in the database. However, these compounds are known to have toxicity and/or stability-related issues and would therefore not be suitable for repurposing. We also noticed that multiple SRC-ABL inhibitors were amongst the top scoring compounds, indicating to us that there was something special about this particular target class, and many of these compounds have been clinically approved. So SRC and ABL are structurally related non-receptor tyrosine kinases that play broad roles in cellular signaling. The SRC family of kinases is composed of 9 members, and most SRC-ABL inhibitors inhibit more than one member of the SRC family to varying degrees. So on the right-hand side, we see a kinoscan tree spot profile for dasatinib. Each of these spots indicates an individual kinase that's being hit by dasatinib, and the size of the spot corresponds to the percent inhibition at a particular concentration of dasatinib. And SRC and ABL-related kinase hits are in blue, and all other hits are in red. And what this is showing us is that dasatinib hits a number of other tyrosine kinases as well outside of the SRC-ABL family. SRC activity has been associated with the pathogenesis of solid tumors, and recent literature has begun to suggest that SRC actually may play a role in regulating the differentiation and activation of myofibroblasts, which are the key pathogenic driver in fibrotic diseases such as IPF. So SRC-ABL-related inhibitors have varying degrees of polypharmacology. Shown here are the tree spot visualizations for 4 different inhibitors, from the most selective to the most polypharmacologic. Corresponding analysis of their IPF potential score suggests that while some polypharmacology may be beneficial, too much could be detrimental. By combining this type of target-based analysis together with phenotypic profiling, we can begin to identify the optimal target selectivity for balancing safety and efficacy in IPF. So we then confirmed the anti-fibrotic potential of a number of SRC-able inhibitors and a more focused BioMAP panel specifically covering fibrosis-relevant biology. This panel consists of 3 systems. The systems cover general myofibroblast activation biology and also include systems containing co-cultures of epithelial cells and fibroblasts to cover physiologically relevant organ-specific biology. The system on the left, SAE-MyoF, is a co-culture of small airway epithelial cells and fibroblasts to model pulmonary fibrosis, and the system on the right is a co-culture of renal tubule epithelial cells and fibroblasts to model renal fibrosis. So this allows us to evaluate the anti-fibrotic potential of these agents in different contexts. And what this data shows us is that this particular inhibitor, saracatinib, has pleiotropic effects. We see a reduction in IL-6 and IL-8 indicating anti-inflammatory activity, but we also see some very interesting context-specific effects on matrix-related readouts. So for example, saracatinib inhibits the expression of alpha-smooth muscle actin, a key biomarker of myofibroblast activation in the fibroblast monoculture, as well in the renal epithelial cell fibroblast co-culture to the right. However, there is no effect on alpha-smooth muscle actin expression in the SAE-MyoF system, which is a co-culture of fibroblasts and airway epithelial cells. So this indicates that there may be differing effects either on its specific cell types or on the paracrine signaling that happens between the two. So when we look at the effect of saracatinib on the expression of extracellular matrix-related proteins such as collagens, we see the same thing. So we then compared the profile of saracatinib in this panel to one of the 2 approved IPF drugs, pirfenidone, and we see some interesting things here as well. In the monoculture and the co-culture modeling renal fibrosis, there are a number of shared activities. However, in the system modeling pulmonary fibrosis, the profile is much less similar. This context-dependent information can also be used to guide indication selection. So here are the profiles of the 4 SRC-able related inhibitors in the fibrosis-relevant BioMAP systems. So we can see here that even though these inhibitors are technically in the same target class, each compound has a very unique profile, likely based on their relative target selectivity. So for example, we see that bosutinib and imatinib increase the expression of IL-6 and IL-8 in the renal fibrosis model, but that bosutinib and saracatinib and dasatinib do not. Saracatinib increases the expression of pro-inflammatory chemokines, MCP1, as well as the adhesion molecule VCAM1 in the pulmonary fibrosis system. And dasatinib is the only molecule that inhibits alpha-smooth muscle actin expression and collagen-1 expression in the pulmonary fibrosis system. So these activities are shared with the approved drugs pirfenidone and nintedanib. So we then ranked the SRC-able related inhibitors based on their relative IPF potential score in the wider panel, as well as the number of shared biomarkers with pirfenidone and nintedanib in the pulmonary fibrosis specific panel. Based on this analysis, we ranked dasatinib as a SRC-able inhibitor with the most potential for IPF. And interestingly, dasatinib actually has recently been shown to be effective in a mouse model of pulmonary fibrosis, as well as able to directly block myofibroblast differentiation in vitro. So this Venn diagram shows the overlap in the kinase target profiles of bosutinib, dasatinib, and one of the approved IPF drugs, nintedanib. Interestingly, all 3 molecules are able to inhibit SRC-able kinases. However, nintedanib and dasatinib, which was the top-ranked hit in our analysis, shared additional targets that wasn't shared with a lower-ranking compound, bosutinib. This includes DDR1 and DDR2. These are collagen receptors that have been shown to control matrix metalloprotease expression, and in addition, both molecules have been shown to inhibit c-KIT, and c-KIT-positive cells in the lung have been shown to promote myofibroblast differentiation in vivo, and this may point to additional targets that contribute to the efficacy in an anti-fibrotic therapy. So to summarize, we've used phenotypic profiling to define the shared characteristics of IPF drugs despite having different targets. We then use this phenotypic fingerprint to search a database in order to identify drugs with similar activities. Using this method, we identified SRC-ABL-related inhibitors as having anti-fibrotic potential, which was confirmed in assays more specifically modeling fibrosis. We were then able to rank these hits based on their overall potential, specifically for pulmonary fibrosis. So one of the biggest challenges for a drug repurposing program is prioritization of potential indications or prioritization of potential drugs. Most companies have a library of drugs that have been through some clinical testing and have been shelved for any multitude of reasons. Given an emerging indication space, such as IPF, how can one quickly identify compounds from the library that might be appropriate for that particular indication? So we all realize that by now that for many diseases, there's more than one way to clinical efficacy, and there's emerging evidence that polypharmacologic solutions may be more impactful for certain diseases. By combining data from human primary cell-based phenotypic screens and data from target-based approaches, one can begin to identify beneficial polypharmacology. And with that, I've reached the end of my presentation. I thank you for your attention, and I think we'll take questions.
Thank you so much, Charlene. That was really great. Let's give everybody 30 seconds or so in case you want to type some more questions, and then I'll start with what we have. Okay, so first question we got was, is it possible to use this approach to look at drugs for something like an orphan indication or a disease for which there's no treatment?
Theoretically, yes, assuming that our systems cover the relevant biology for the disease in question. However, one thing I wanted to point out is that you don't know what you don't know. So for example, we saw shared effects of pirfenidone and nintedanib that have not been currently characterized as playing a major role in IPF, and some that have. So these activities may indeed come to be important for clinical efficacy, but only time will tell. So the strength of phenotypic profiling, I think, is that you can help get information on the downstream effect of multiple targets. But theoretically, yes, it could be used, assuming that, again, these systems cover the biology of the disease in question.
Thank you. Next question is, if you were specifically interested in IPF, why couldn't you have done your initial evaluation and scoring in the smaller fibrosis panel that you showed?
So our reference database actually covers a larger diversity plus panel, but this example is also sort of meant to demonstrate what somebody would want to do as a real-life example, as a potential screening strategy. So ideally, if someone has a compound or a library of compounds that they want to repurpose, they would want to cast a wide net in terms of potential indications, right? So starting with a broader screen means that you could potentially evaluate multiple indications at the same time, and then sort of have a funnel strategy to follow up on the most promising indications.
Very interesting. Okay, so it looks like we just have one more question for you. Multi-targeted drugs may have increased efficacy, but may also have increased adverse effects. How could this be addressed in this type of approach?
So that's a good point. One of the flip sides of polypharmacology is sometimes increased clinical adverse effects. And the scoring system that we used for this particular example was very straightforward, really only incorporating the shared effects of the two approved drugs. However, this scoring system could theoretically be expanded to penalize a profile if they included readouts related to certain adverse effects, assuming that there was a correlation to certain readouts and certain adverse effects. You could easily modify the scoring system to reflect that.
Very cool. All right. Well, if there's no more questions, I'd like to close it there. Just a quick administrative reminder that you should be receiving the slides and a link to this webinar tomorrow. And in the interim, if you have any questions at all, please don't hesitate to reach out to us. Thank you all for joining, and we hope to see you soon.